Hello, and welcome to Level 3. My name is Rio, and I will be helping you continue your journey in learning about cells. In this video, we will be learning about microbes, what they are, why we know so much about them, why computers play a surprisingly large role in how much we know about them, and how they affect our day-to-day lives. I hope you enjoy. As you have learned in the Level 1 and Level 2 videos, cells are a fundamental part of every living thing we know. We can think of cells as the building blocks of all living things. They are the cells that you are made up of: nerve cells, skin cells, red blood cells, white blood cells. Cells make tissues, tissues make organs, and organs create organ systems that make you. While the cells that make up your body are very important for humans, and honestly, also important for every other multicellular organism in the world, we are going to look at the cells that choose to live a more independent lifestyle. These single-celled organisms include bacteria, protozoans, fungi, and even certain types of plants that live all around us, in the air, on surfaces, in deep-sea vents, and even in our bodies. To classify all of these different types of living things under one umbrella, we will call this group of single-celled organisms microbes. It is also important to note here that viruses, while also considered a microbe, is not a single-celled organism. Viruses, such as coronavirus, are quite different from cells and will be a story for another time. While it is quite easy to see why the cells in our body are important to us, it is harder for us to appreciate microbes. When you think about microbes, you might think about small, boring little blobs that have little impact on our daily lives. In fact, the only time you ever hear news about microbes is when people talk about how dangerous they are. We have antimicrobial soaps that we wash our hands with, and hand sanitizers that remove 99.9% of germs. So microbes must be bad, right? But in fact, the real story is that the cells that live around us are a diverse group of organisms that have shared a complex relationship with people. Microbes are just as important to our lives as the cells that make up our bodies, and even in modern times, these microscopic, single-celled organisms continue to subvert our expectations in fascinating ways. Before talking about the many ways microbes impact our lives, we must first talk about how we know so much about our microscopic neighbors. Since the discovery of the first cell, the most effective tool that biologists have used to learn more about microbes is the power of observation. Previously, we've discussed three fundamental ways we can use to observe and understand the cell: physically, chemically, and biologically. When Antoine van Leeuwenhoek, the father of microbiology, first viewed microbes under a simple light microscope, he was learning about these cells through physical observations. Leeuwenhoek even created illustrations of what he saw and could make out the detailed structure of the cell. However, while Leeuwenhoek's work was extremely important in starting the study of microbes, his observations were limited by the technology of the time. Humans have been observing cells for a long time, and our methods of observation continue to get better and better. While the first cells were observed using simple microscopes, new techniques and technologies have created an explosion of new information about our microscopic neighbors. Physical observations have greatly increased with the introduction of new types of microscopes, including the scanning electron microscope, SEM. SEMs have magnification abilities that are 10,000 times more refined than the microscopes that Leeuwenhoek used, and SEMs can be used to view the world of microbes with incomparable quality. Chemical reactions are important drivers for functions of cells and microbes, and chemical observations have become much more accessible in the last 60 years. It is now possible to map out the many chemical reactions that occur in all cells, which allows us to track how microbes respond to certain factors such as antibiotics. Finally, the advancements of genetic sequencing, or the process of deciphering the biological code that programs all organisms, has exploded the rate at which we can perform biological observation. The advent of various techniques, such as next-generation sequencing, created by the company Illumina here in San Diego, helped greatly increase the speed and efficiency of sequencing genetic material. This technology has allowed your average researcher to perform sequencing experiments that would have taken 10 years to accomplish in the 1990s to be done in a matter of days. And with greater ability to decipher this code, we continue to better understand how life has continued to persist on this planet. However, while the improvements in measurement technology has greatly improved our understanding of the microbe, there has been another technological advancement that has allowed us to understand these microorganisms: the computer. While the connection between single-celled organisms and computers might seem like a weird jump, let's try a simple thought experiment. For the purpose of this exercise, you need only know 3 things. First, DNA is the code that gives different species and even different members of the same species its features. Humans have very similar DNA codes to chimpanzees, but human DNA has an even closer match to another human, even if we think that 2 people look very different. Second, DNA is made up of 4 different building blocks, and when sequenced, we simplify each building block to a single letter: A, T, C, and G. Both the order of the building block and the building blocks themselves are important to give an organism its features. For example, ATT will have a very different effect on the organism compared to TTA. And number 3, mutations can occur in an organism, which is what happens when some part of the genetic code is changed. This can happen for a variety of reasons, but even a change in a single building block can result in huge shifts in how the organism looks or behaves. So, now on to the exercise. Imagine you are a scientist and you have a partial genetic sequence for 2 microbes of the same species, but one cell has a mutation. Sequence 1 represents a normal microbe, while sequence 2 represents a microbe that looks the same but can glow in the dark. It is your job as a scientist to find a mutation between the 2 sequenced DNA samples. Can you spot the one difference between the 2 sequences? Feel free to pause the video to give yourself more time. If you are able to find the differences, you are a master puzzle solver. However, for most people, myself included, the work of shifting through all these A's, T's, C's, and G's to find the one small difference is a monumental task. And this is a relatively small chunk of DNA with a length of about 650 bases or letters. The average microbe will have an average base length of 4.4 million bases, while the average human has a DNA that contains 3 billion bases. Even for a team of well-trained humans, the task of finding the differences in DNA between every cell would not only be a difficult task, but an impossible one. This is where computers come in. Computers are excellent at processing large volumes of data using repetitive processing techniques, and will execute them to a level of accuracy much greater than your average human. In fact, the power of computers have become so impactful in the field of biology that there is an entirely new field that has been created out of it, the field of bioinformatics. In our Level 2 video, we discussed how scientists construct models of cells using real-world data so that we can have controlled, consistent, and detailed understandings of our microscopic neighbors. And while the new observation techniques, such as electron microscopes and next-generation sequencing, are able to produce large amounts of high-quality data, it is computers that allow us to combine all the information into something accurate and understandable. For example, bioinformatic researchers have used techniques such as X-ray scanning to create detailed 3D models of unique molecules that can be found in cells and microbes. These unique molecules, called proteins, serve an important role in microbe survival. Understanding their structure allows us to fight bad microbes or help good ones. However, such models are only possible with the help of computers to process the data and convert it into something we humans can understand. With a combination of new observation techniques and increased computing power, humans have gained incredible understanding about the invisible organisms that live everywhere around us. In fact, our understanding of microbes have allowed us to make some pretty incredible changes in our day-to-day lives. And here's how. So far, we have discussed the many tools we humans use to learn about microscopic organisms. These tools have given us a much deeper understanding of the physical, chemical, and biological aspects of the cell, and over the years, many impactful discoveries have been made. One such discovery was the identification of the microbiome. Previously, we mentioned how the only time you are likely to hear about microbes is when talking about how dangerous they are, because they cause diseases. And it is true that some microbes can cause diseases. However, microbes that cause diseases, also known as pathogens, make up only a tiny fraction of all species of microorganisms. only 1% of known bacteria can infect humans and make us ill. For most microbes, they choose to live their lives without causing harm to a host, living on various surfaces or even in the air. And on the flip side, microbes can even provide a direct benefit for us. We humans have a large population of microbes that live on and in us. From your skin to your mouth to your gut, it is estimated there is an almost 1-to-1 number of microbe cells to human cells that make up you. And while that might sound pretty gross, these collections of microbes, also known as your microbiome, are often vital to your health. For example, certain gut bacteria can allow humans to extract nutrients out of foods that would otherwise be indigestible, allowing us humans to eat a broader range of foods. There have even been several studies that have found how shifts in the microbiome are correlated with certain diseases. This means that it is possible to map out changes in your microbiome as a diagnosis for these diseases. So, while it is still important for you to wash your hands to avoid catching any bad pathogens, to say that all microbes are bad is simply just false. The tools that have helped humans better understand microbes have also been invaluable in changing our daily lives. Armed with our knowledge about microbes, humans have found that many single-celled organisms can be used to help create everything from food to vaccines. While we have already explained how microbes such as yeast help make our breads fluffy, there is a huge range of other industrial applications for our microbe friends. Microbes are genetically engineered to produce a desired product. By modifying the genetic sequence of a cell, it is possible to use the cell's own machinery and chemical reactions to create complex molecules. These newly genetically engineered microbes are then grown in large quantities and the desired product is extracted from the cells. Using microbes is especially useful for building complex molecules such as medicines and proteins because cells are already well equipped with the necessary chemical reactions to do so. There are a huge variety of possibilities that can be created using genetically modified organisms, and people have already found many unique and interesting applications. In 1982, the first ever medicine that originated from a genetically modified organism was a microbe-derived human insulin, marketed under the name Humulin. Insulin is a vital medication that is used to regulate blood sugar in diabetic patients, and the arrival of Humulin has helped improve the quality of life for many people. In more recent times, a company named Air Protein is working to use microbes to take the animal out of meat. The company states that it will be using specific microorganisms to convert CO2 in the air into a protein powder, which can be then used as a meat alternative. The possibilities with microorganisms are endless. These little micro machines have already become an important part of our lives, and they will continue to solve many of the issues that humans face in the near future. And now, dear viewer, armed with a greater understanding of the microscopic world, you may one day use your knowledge to help make this world a better place. Thank you for watching. Hope you enjoyed it. We have one last video, level 4, where you will hear from some actual microbe researchers on their own research, college advice, and why they love to do what they do. Hope to see you there.